Yeah. Right, everyone. Thanks very much for coming. I think there's tea and coffee outside if you wanted to grab one before we get started fully. I'm Anna Murray. I'm the Director of Research and Impact for Clinical and Biomedical Sciences Department, and I'm going to be chairing today and introducing our speakers. So we've got a few housekeeping things just to start. We have a slide on this. Yeah. Oh, just. So, yeah, before we get to this, just to say that there are fire exits out of this door to the left and to the right as you come out of that door. The nearest toilets are out here on the left. So there's tea and coffee, and there will be lunch at 1 o'clock outside here. And we're going to be recording this seminar. We've got 3 speakers today, and what we're going to do is take questions after each of those people. So a couple of other things to mention before we get started. First of all, to remind everyone that we, as members of the BRC, should be acknowledging the BRC on any outputs coming from your research. And this is the statement that you should be using on that acknowledgement. And obviously that's on the website as well. And also to remind you that there's a deadline this Friday for a pre-application support fund. So this is a small pot of funding to support healthcare professionals who are going for these NIHR doctoral and advanced fellowship opportunities. And there's some more details here. So if you're going for that, just to remind you that the deadline is the 15th of November, which is this Friday. And another thing to bring to attention is the university have just launched this Pivot RP facility, I suppose, and it's replacing the research professional. And this is a sort of a website or tool that you can use for accessing funding opportunities. You can set up searches and reminders, and it's a really good way to do a big search of the type of sort of funding opportunities that there are out there. And there's various training sessions available if you want to have a look. I think it's fairly intuitive, though, that you can just go on and have a go and use it. So to remind you that that's available. So on to the sessions today. As I said, we've got 3 speakers. Each person will have, Nick and Sonia will have 20 minutes to speak, and then 5 minutes of questions after each of theirs. And then Emily will have 5 minutes to present her work and 5 minutes for questions. And then that should take us to the hour. So we're going to start with Nick Kennedy, who is a consultant gastroenterologist at the hospital at the RD&E, and also a senior investigator fellow at the BRC. So Nick, take it away.
Great. So thank you, Anna, and thanks to the BRC for the opportunity to speak today. It's nice to have plenty of you in the room as well as online. So I'm one of the consultant gastroenterologists. I've been in Exeter for 9 years, and I've had an honorary association with the university for all of that time, although become a senior investigator fellow of the BRC when those opened a year or so ago. I'm going to talk about pharmacogenetics. So this has been a big part of the work done in the Exeter IB research group, which I became part of when I came here. We've been interested for some time in trying to better understand how to use the various medications we have to treat inflammatory bowel disease, and how genetics might be used as a tool to predict response as well as adverse drug reactions. Inflammatory bowel disease is an increasingly common problem. The main 2 types are Crohn's disease and ulcerative colitis. And collectively, they affect about 1 in 140 of our local population. We did some prevalence work about sort of 7 or 8 years ago, looking at this in primary and secondary care, so we know roughly where we are. But because of although the incidence is now relatively stable, the compounding effect of prevalence, because it's often diagnosed when people are younger, is that the prevalence is increasing, and we will hit 1% in the next decade or so. So not as common as diabetes or heart disease, but an increasingly common problem among our population. We have lots of medications used to treat this disease. And as I said, it's then important for us to be able to use biomarkers to be able to understand how people are going to respond to them. I've got some disclosures. I have done work with the pharmaceutical industry over time. So pharmacogenetics is the study of variations in genetic sequence as related to drug efficacy, dosing, and side effects. And the idea here is to maximize benefit of drugs, minimize harm, and reduce costs so that we give people the drug that's most effective for them. In practice, certainly in the IBD space, predicting drug response has been much harder than predicting adverse effects. And I think there are a number of reasons why that might be. I think drug response is a much more complex phenotype. There are lots of other things that contribute to why people might respond or not respond to a drug, including variation in the underlying disease, as well as the way an individual processes the drug and the way their immune system functions. Whereas adverse drug reactions tend to be more similar among individuals who have them. We've done more work on the adverse drug reaction side than we have on the prediction of response. We've been working on this for quite some time, including before I came to Exeter. And I just wanted to sort of highlight that the journey for biomarker discovery to clinical implementation, not just for pharmacogenetics, but in general, is quite a long process, and typically said to be about 17 years from initial discovery of a potential biomarker through to proving its validity, utility, cost effectiveness, and then clinical implementation. In the pharmacogenetic space, the rapid drop in cost of sequencing has made it much easier to think about doing large-scale genomic work. This slide's obviously slightly out of date now, but this was when the whole genome sequence cost first came down to $1,000, and it's come down further since then. And it then becomes a really important problem to define the phenotype once the genetics becomes a relatively low-cost thing and relatively straightforward in terms of the samples needed. Defining clean phenotypes that allow us to split people into cases and controls becomes a really important task. Tariq Ahmad, who's one of the other key members of the team and predates me in Exeter, who sort of set the group up, has been the key person driving a lot of this work and is also associated with the university here. He set up a network of 140 sites around mostly the UK, but also internationally. So there's some in Canada and Israel and Australia, for example, trying to build cohorts of cases with various different types of phenotype around drug response, and then getting either blood-based or salivary DNA to do genomics. And since I joined the unit 9 years ago, we've worked closely together with Claire Boucher, who project manages the work, and James Goodhand is one of the other gastroenterologists, and a range of fellows over time on this. So we had clean definitions of phenotype, and we used a web-based tool for gathering the data from all of these different sites from around the country and the world, and then worked with genomics laboratories in the UK and internationally to do a mixture of genotyping and sequencing. One of the key steps for this is adjudication, and this has been a really good way of involving a broader team of clinicians in this work. So this is, once we've got the case reports from the various sites, we have then sat around a table, although some more recently has been done virtually. It traditionally has been done with bringing everyone to Exeter, including people from elsewhere around the country, and discussing the cases. So we look through the cases individually, we make our own assessment as to whether something is a definite probable, possible, or unlikely adverse drug reaction. And then with 2 or 3 of us looking at each one, we then bring that together in discussion to try and have a final adjudication. And that, I think, has been a really important part of ensuring we have a clean phenotype to test against our genetics. So this is the schema that we followed. It's based on a paper from 2011. Which allows us to split people into these 4 categories. And so to have a definite drug reaction, there has to have been a re-challenge that was positive, but we can also have probable or possible ones. And for most of the studies we've done, we've then focused on the probable and definite as being the cases. And then for controls, we've taken people actually mostly from our local IBD population who haven't had a drug reaction and where we've got good data to support an absence of a drug reaction. Thiopurines have fallen more out of favour probably over the last 5 years as the cost of advanced therapies in IBD have come down, but they do remain an important part of inflammatory bowel disease management. They've been around since the 1950s. The people who discovered thiopurines ultimately won the Nobel Prize, although for some of the other drug discovery work they did. And they originally described mercaptopurine in 1953, and then went on to develop azathioprine, which was described about 1956-57 as a prodrug that had more predictable bioavailability than mercaptopurine. And they've historically been prescribed in over half of patients with Crohn's disease and about 1/3 of ulcerative colitis within the first 5 years of diagnosis. However, adverse drug reactions to thiopurines are common. These include myelosuppression, so low white cells, sometimes associated with infection and sometimes serious infection, liver injury, pancreatitis, thiopurine-induced hypersensitivity, which is typically early drug reaction, and people get fever and joint pains and other such things. And then probably the most common, but the least serious of these is gastrointestinal intolerance. And so we've actually been doing work on 4 of these 5 from a pharmacogenetic perspective. Thiopurine-induced pancreatitis was the first of these that we published results, and this is an important outcome in that it affects people affects about 3 or 4% of people overall treated with a thiopurine, and it often leads to hospitalisation, although it does usually resolve when the thiopurine is withdrawn. And so we were able to do a genome-wide association study identifying a location on the HLA associated with this outcome. And interestingly, when we first published this, the view was that this probably wasn't quite good enough to use it as a pharmacogenetic test, because it would preclude a reasonably high number of patients from receiving thiopurines. But actually things have moved forwards, and I think now there's a much stronger case to use it where there's many other options to choose instead of thiopurines, and a 17% risk of pancreatitis among those who carry the relevant HLA variant is quite high. More recently, we also worked on myelosuppression, so this is low white cell counts in the context of thiopurines. This is something that was perhaps one of the earliest clinical uses of pharmacogenetics, not driven by us, but driven by others, related to thiopurine methyltransferase. So this is an enzyme involved in the metabolism of thiopurines, been known about for a long time, and patients who have low levels of TPMT activity get high levels of the therapeutic metabolites, which lead to toxicity because they cause suppression of white blood cells. So that's been known about for a while, and it's one of the earliest pharmacogenetic tests to come into clinical practice, and it's widely done, certainly in the UK, but across the world, for patients about to be treated with a thiopurine. We wanted to look beyond that, to look for other variants, because there was still an increased risk of myelosuppression despite TPMT testing. And we were able to discover some additional NUTD15 variants that were associated with a much higher risk of myelosuppression in Europeans. This gene had previously been associated with, variations in this gene have been associated with an increased risk of myelosuppression in East Asians. And we found that we were able to show that as well, but also in a European population for the first time, we're able to show that an insertion deletion variant was associated with this outcome. And putting this together with TPMT, we're then able to show that you can predict with some reasonably high certainty in the people who carry variants in both that people will develop myelosuppression. We've done some additional work since then in a larger population-based cohort as part of the NIHR IBD bioresource. So this is a national study that now has almost 50,000 patients with inflammatory bowel disease. It's hosted in Cambridge, but Exeter has been a key participating site in the bioresource since its beginning, and I've been involved since the beginning of it as well, along with other colleagues here. And so we were able to use a phenotype-first approach here, and then, a genotype first approach here, so going from genotype to phenotype to show, again, an increased risk of myelosuppression, but also severe myelosuppression, so very low white counts or neutrophil counts, and myelosuppression related to hospitalisation. And for both of these two more severe outcomes, the odds ratios are considerably higher. We have done this work and have been able to continue to get further data around NUTD15 in this population, both in Europeans but also South Asians. Actually, our initial going into this, we thought we were going to be able to show that this was a test that should be offered to everyone. We've been working with the test group here in Exeter on the cost effectiveness, and really for South Asians and East Asians, it's relatively straightforward, I think, to justify genotyping people ahead of treatment, and you don't need to genotype many to prevent an episode of myelosuppression or even severe myelosuppression. In Europeans, while there's still clinical value in doing this, and it is still an important cause of myelosuppression, the variants are much less common, and the cost effectiveness argument is much harder to justify. So at the moment, I think we're going to be heading towards trying to get it onto the test directory for people of Asian ancestry rather than European. So just to talk about some of this overall journey, we've gone from having some initial publications back in 2014, 2019, through some focused reverse phenotyping, as well as confirmatory testing, which I haven't discussed in the clinical laboratory here. And I think one of the successes of our work has been close collaboration with both the clinical genetics and clinical biochemistry laboratories in Exeter. We've worked with the Exeter test group around cost effectiveness, and the aim is then to get some of this into the genomics test directory so it's available for clinical use. One of the other big projects that I've been involved in since I came to Exeter is the PANT study. So this is a large study of around 1,500 people from around the country treated with an anti-TNF drug for the first time for Crohn's disease. So this is either infliximab or adalimumab. We actually went in to do a pharmacogenetic study of both drug response and other outcomes related to these drugs. Actually, drug response, as I mentioned earlier, has been really hard to predict, and even actually extending beyond genomics to looking at methylation and transcriptomics and protein and glycomics, which we've done within this cohort. We've been unable to find any really good predictors of drug response, but we have found a genetic predictor of immunogenicity. Again, this is in the HLA, so an HLA variant in so the HLA haplotype of DQA1*05 is associated, independent of concomitant medication, with an increased risk of forming antibodies against these drugs. And this is clinically important because the antibodies go on to block the effectiveness of drug over time. These graphs show that when we look at the genetic variant on the side, looking at treatment with thiopurines, which can be used to modify the risk of antibody formation, we see this in sort of additive effect effectively. And for infliximab in particular, if you're unlucky enough to both carry the HLA variant and not be treated with a thiopurine, almost everyone develops some antibodies within the first year. This is something that we're probably a little bit further away from getting into something like the genomic test directory, but actually has been taken up internationally and to some degree in the UK as well. And the people are doing this as a clinical test and using it to inform treatment decisions. But I think some of the, we're not quite as far down the route in terms of cost effectiveness work as we are perhaps for the NET15 testing. Briefly touch on some of the other work we've been doing. We have also done, as I said, thiopurine hypersensitivity and liver injury projects. These have taken us a bit longer to get to completion. Liver injury in particular is a more complex phenotype in that there are different patterns of liver injury that can be associated with thiopurines, and I think that's made it harder to pin down exactly what's going on. We did a really big pandemic era study. We recruited 7,500 patients from 92 sites around the country with Crohn's disease or ulcerative colitis who were being treated with biologic medications, and looked at the development of anti-SARS-CoV-2 antibodies, so exposure to the infection, and then with the rollout of the vaccination programme in late 2020, early 2021, we went on to look at the effect of the medications on immunogenicity effectively driven by vaccination. So in that case, you want antibodies obviously after you've been vaccinated. And we did some important work there showing a reduction in antibody responses to vaccination and resulting probably from that higher rates of breakthrough infection in patients treated with infliximab if they were vaccinated against SARS-CoV-2 than if they weren't on infliximab. And this was used to inform national and international guidance around additional vaccination in those cohorts. On the pharmacogenetics side, we're then keen to use the overlap between our Clarity IBD cohort that I've just described and the bioresource to look at the pharmacogenetics of vaccine response. So we've got about 3,000 patients who were both in the IBD bioresource and in Clarity IBD, and we're working with colleagues in the Sanger and elsewhere and in Exeter on a pharmacogenetic project. It's been a bit delayed by some sort of logistical issues, but we're hoping we can get that moved forward soon and identify some useful things, because these are likely to be relevant to other vaccinations as well, not specifically just SARS-CoV-2. The PANT study I mentioned before, we had a number of useful clinical insights as well as the pharmacogenetic one I've highlighted here, and we're now working on a novel clinical trial that takes one of those further forwards, and we're hoping to apply for funding from NIHR and others over the next sort of 6 months or so, and I'm starting to work with clinical trials units So there's plenty going on, and I think it's been an exciting team to be part of, and it's really good to have be starting to form links across the BRC with others interested in the genomics of complex disease. That's all I had to say today. I'm happy to take any questions. Are we doing questions from the people online as well, or just from? Yeah, that's great.
Yeah.
So thank you very much.
Are there any questions in the room for Nick? Oh, yeah, at the back there. Hang on, can you wait for the microphone for a sec? Sorry, thank you. Just so people can hear.
Hello. Oh, hi. So I was just wondering, with regards to the NUDT 15,
Yeah.
do you think that's applicable to the use of mercaptopurines in other disease cohorts?
Yes, very much so. So for the most part, pharmacogenetics of adverse drug reactions are likely to apply to whichever disease they're used in. Clearly, there will be for diseases that at least have a genetic component, which inflammatory bowel disease does. There may be a difference in the genetic background of the individuals being treated, which could influence the impact of genetic variants on adverse drug reactions. There may also be differences in dose. For example, the captoprurine is often used at considerably higher doses to treat acute leukemia than is used to treat inflammatory bowel disease, and that may interact with the genetics to determine one's risk of an adverse response. There is already some approval for NUDT15 testing in the haematology setting within the UK. It doesn't extend beyond haematology, so that's come about actually after we originally described this variant in Europeans. But yeah, it's likely to apply to whichever condition they use to treat. I think both in terms of TPMT testing, which has been around for a long time, but also in terms of use of thiopurine metabolites as another tool used to guide dosing. I think it's fair to say that the IVD doctors have been more proactive at taking those up than has been the case in rheumatology, dermatology, or even hepatology. That's a related field.
Great. Yes, I work in neurology, and we use it quite a lot for myasthenia, and we tend to do TPMT, but that's pretty much it in terms.
Yeah, it's good because we TPMT, and I think NUDT15 variant testing. When you can do it now, there are labs that will do it for you if you prepared to pay, but it's as I said, because it's not in the test directory outside of leukemia, it's limited perhaps uptake in the UK. But the latest round of the CPIC guidance, which is the international sort of pharmacogenetic guidance, is coming out soon on this, and we're hoping to align both that and the UK NHS test directory around its use, at least in the East and South Asia population.
Great, thank you.
Chris, there's a question over there. Thank you.
Thanks, Nick. That was really interesting. I was just wondering if you could give us any indications about the mechanism of the interaction. So what is NUDT15 doing?
So it's involved in the metabolism of thiopurines, as TPMT is. TPMT is better understood in that setting, in that when we draw up so thiopurines can be metabolized through 3 different paths. They can end up as thiuric acid, which you pee out and doesn't seem to cause any harm, but isn't terribly useful either. They can go to the methylated captoprurine metabolites, which are associated with liver toxicity and aren't helpful. And then the 5-guanine nucleotides, which are associated both with the therapeutic effect because they interfere with DNA synthesis, but also with myelosuppression. And so if you inhibit TPM, or if TPMT is deficient, you get less of the methylated methylated metabolites being formed and more of the 5-guanine nucleotides, but you end up with too much of them, and then you get the myelosuppression effect. So you can get around that by dose reduction. NUDT15 is also involved in that metabolism. It's more complex, but it looks as though you probably can use reduced dosing of thiopurines in people who have NUDT15 variants to mitigate some of it. Although I think it's less straightforward using dose reduction in that population probably than it is in the TPMT intermediate group, where we're very used to just having a reduced dose in the 11% or so of people who carry a single TPMT variant.
So just as a follow-up, does that give you the potential for rational drug design where you could create a drug that can't be metabolized in a certain direction?
That's a really good question. I mean, I think in this case, the problem is that so yes, potentially, and it also raises, I guess, the possibility of using 5-guanine directly, because these are, I mean, both the captoprurine and thiopurine are effectively prodrugs. You're giving them and then expecting the body to metabolize them. And it's this inter-individual variation in metabolism which leads to problems. Phytoadenines are occasionally used, but we've not used them in Exeter as far as I'm aware, but there was a fashion for using them, particularly in some of the London hospitals. There were some concerns about a sort of specific side effect with those, I think related to the liver, but I might be wrong on that, which I think has never been fully substantiated. But I think partly also because of the rising availability of cheaper advanced therapies, there's been had to move away from thinking about thiopurines quite so much. And thiopurines are interesting in that they are an example of rational drug design to a degree, and a relatively early example themselves, in that they were the intention of the biologists who were developing these was to interfere with DNA synthesis, and they were testing a whole series of molecules that were similar to purines, so they're purine analogues, to see if they could do this, and looking at differences between tumour and healthy cells. So they're perhaps not in quite the same way as, say, imatinib, where someone identified the Philadelphia chromosome product and tried to screen molecules to target that specific thing. But they are perhaps a sort of reasonably good early example of not just throwing a drug at a disease, but trying to think about ways of interfering directly with biology using drugs.
Thanks. Are there any questions online from anyone? See any? Can't see any hands up. Nothing in the chat. Okay, I think we should probably move on then, Nick. Thank you very much. Thanks. You didn't have your acknowledgement slide. That is his acknowledgement slide.
Yeah, so just to point out, I mentioned earlier about some of these other people, but it's like I'm sure all of you experience, you can't do big projects like this without a lot of good people around you. And we work closely with the lab, with the research nursing teams and others to make this happen.
Great, thanks. Yeah, important to mention that, isn't it? Okay, great. So our next speaker is Sonia Medina-Hernandez. And Sonia graduated with in neuropsychology in Madrid, did an MSc in psychological research methods, and then a PhD in neuroimaging at King's College London, and came to Exeter in 2023, not long ago, and is a BRC fellow. Talk to us about your work, Sonia. Thank you.
Thank you. Hi, can everybody hear me okay? Okay, cool. I can use this. Perfect. Well, thank you for inviting me, and thank you everybody for coming. As you were saying, I've been here for like just about a year and a half, doing like research about chronic pain, not involving necessarily chronic back pain patients, but in healthy individuals. But today I'm going to be talking to you about the first project that I'm doing as far as BRC. I only started this 6 months ago, which means that unfortunately I'm not going to be able to show you any data, but hopefully I'm going to be able to provide you enough context and just tell you about what I've been up to for the last 6 months. The project is called Mind the Gap, Steps Towards Precision Rehabilitation for Chronic Back Pain. And this whole Mind the Gap and training innuendos will make sense later on in the presentation. But for now, just going to give you a little bit of context, just a bit of numbers. Chronic back pain is a global health crisis. The International Association of the Study of Pain, as of 2021, said that lower back pain still is the leading cause of years lived with disability since the 90s, and it's not really getting any better. Currently, there are about 557 million people around the world living with chronic pain, chronic back pain. And this is not really because the scientific community is not trying to catch up with this. If you do a quick search on PubMed, you will see that only in 2024, there's almost 800 studies looking at chronic back pain therapies, for example, or over 100 studies looking at chronic back pain and mechanisms of it. Yet nothing really seems to be changing. And this is to do with, I'm not sure if you can see that very well, but this is to do with what we called, or what I call anyway, the gap. There seems to be a gap. And if you look at one side of that gap, you have all the, research, neuroscience research, clinical research, basic research, and we have keeps growing, it's ever growing. Like we continue creating new sources of knowledge and truth in animal research, human research. Obviously, artificial intelligence is flourishing in the last few years, a state-of-the-art neuroimaging method. So we are able to capture neural dynamics by the order of milliseconds and within millimeters of the brain. And now we've seen a really good example of, genetics and pharmacology and both together. But then when it comes to chronic back pain, or when it comes to chronic pain in general, patients feel that they're still very much stuck in the past, right? So they get offered treatments that give them little to no pain relief. They also report a high lack of patient-clinician rapport. So they don't really feel like they're being understood by their clinicians. They don't really feel that they're being trusted, and that in turn provokes them not to really have trust in the system, in the health system. This, as a lot of us would know, there's a massive translational neuroscience crisis. So a lot of the stuff that gets done in the lab doesn't really make it to like real life scenarios, or it doesn't really make it to like applications that patients can actually perceive. And that all that together leads to an incomplete mechanistic understanding of chronic back pain. We don't really know why it happens in a lot of the cases, and we don't know why it just won't go away. And obviously, and as we all know, the opioids crisis that we are all aware of, and there continues to be a massive issue. So this translational fellowship arises as an attempt to sort of bridge the gap rather than working on one side of the gap or the other, but sort of like put those two together. But first, I guess we need to understand why that gap exists in the first place. And the three main topics that come to mind to me when I think about this gap is first that experiments do not recreate real life struggles that we are aiming to treat, and we'll talk about that later a bit better. Then there's a huge research patient-clinician disengagement, as I was just saying and introducing to you. And lastly, there's a huge lab-to-bench translational timelines. So this, or what I've done so far, what I'm going to talk about today, and what this fellowship is going to focus on is probably the first two. But now I'm just going to talk a bit more about each one of them, hopefully convince you that this is true. So first of all, experience will not recreate real life struggles that we're aiming to treat. And the concept that is really important for everybody to keep in mind is that pain is context dependent, right? So when somebody experiences chronic pain, that pain would be exacerbated or would minimized or would be triggered by experiences that people deal with in their real lives, experiences that are particular to each one of these people. And this could be, picking up your kid or cleaning the house or commuting in public transport. So it is really important for us as scientists that if we are trying to understand the mechanisms underlying pain in these situations, we need to take that context into account. However, the experimental designs that we use to understand those mechanisms that depend on those contexts look more like this, right? They resemble nothing that has to do with real life scenarios, nothing that these people would have ever gone through in their normal daily lives that triggers their pain. And a lot of the time it doesn't, it's for humans, so, which, it has been useful for a lot of things, but somewhere along the way, we're missing the mark now. And, I want to introduce a couple of theoretical models to, hopefully explain a little bit better why context is important. And the first model just very briefly, the first model that I'm going to talk about is a fear avoidance model of pain. Under this model, what they postulate is that when there's an injury, you could go down two main paths. So one of the paths would be you confront that pain, right? Like you continue doing the things that make you feel good. You continue doing rehabilitation. You continue trying to face those symptoms and know them better. And that very usually sets you off down the path of recovery. However, if you are a person, especially if you're a person who's like psychologically or affectively vulnerable, you might start catastrophizing about your pain. And what this means is that you might start creating negative expectations, like excessively negative expectations about what is going to cause you pain, how much pain is going to cause you, what is the disability that's going to come from that pain, et cetera, et cetera. What that leads to is pain-related fear. So you start fearing things around you, and that is going to lead you to the hypervigilance and avoidance of behaviors. Now, we know that with those avoidance of behaviors and being withdrawn around, about things that you used to be able to do and you're not doing anymore, due to that fear, not due to the pain, that provokes, substance misuse, depression, further disability. And what we know, of course, is that increases the likelihood of having further injuries. Now, the important thing to take into account here is that whether you go through one path or the other is very usually not a conscious decision. So by the time that you realize you've been on one side or the other one of the scale, those symptoms are already very well established. So it's really very important for us to be able to pick up early signs of whether one person is going down to one path or another, to be able to sort of break that cycle early enough and not, set this person into this vicious cycle they can't really get out of. And the next model I want to talk about is how, this pain-related fear, exacerbates pain. And this is the Bayesian predictive coding model of pain, which is a bit of a mouthful. And essentially, under the assumption of this model, what you see in the panel A would be like your textbook postulation of this model, which says that for pain perception to arise, there has to be certain elements. So the first element that you have is, do I have a pointer? It's okay. So the first element that you would have would be your prior expectation. So in red over there, the prediction. So you would have a prediction, depending on the context, a prediction of how much do you think something is going to hurt. Then on the other side, on blue, you would have your sensory input, right? Like your sensory system would tell you, give you an approximation of the intensity of the stimulus of within whatever context you are. And the difference between that sensory input and that prior expectation would give you a prediction error. Now, the purpose of the brain analytics model is that to minimize that prediction error. So your final perception would arise as a linear combination of these two distributions. Your prior expectations are your sensory inputs. What happens is that if you are a person who has gone through this pain-related fear and pain catastrophizing cycle, your prior expectations, when you see the number B, your prior expectation might be skewed towards the right, so it's like a lot of pain, and you might be really certain that that pain is going to come and it's going to be bad. And therefore, the sensitivity of your sensory system is going down at the same time. So you don't really have a very good indication of the actual intensity of stimulation that's incoming. And therefore, your final perception is skewed towards your excessively negative expectations. And this cycle just gets stuck and you can't really get out of it. And in this context, all these, which I'm not sure you can read here, but like on the top, you can see that all these fear learning conditioning and these fear patterns and this catastrophizing and this negative effect would mean moderators of this particular phenomenon. So this is all to say that we really need to understand what's going on when people with chronic pain learn fears about certain situations in their daily lives. So what we decided to do was take people who have chronic back pain as well as age-matched health controls. And the first thing that we can do is we haven't done yet, is just have an initial idea of what, a psychometric assessment of what their negative effect and their level of pain catastrophes are. And then we're going to do a good old fear learning conditioning protocol, where there's going to be a fear conditioning phase. We're going to present a conditioning stimulus, that in this case would be an auditory cue. And that will be followed by an aversive stimulus. And we are going to test whether people can, people get conditioned to fear this auditory cue or not. We're then going to have a test phase to make sure that this works. And then towards the end, we're going to have an extinction phase to look at, whether people can extinguish these fear-related responses by presenting the cue, the auditory cue, but not the aversive stimulus. And all of that would be alongside repeated measurements of pain thresholds to look at whether, differences in fear conditioning affect pain. Now, if you just look at this has been done to death in people with chronic pain. So, why doing it again? Well, because usually when people do these kind of experiments in the lab, usually the cue would be some sort of light or auditory cue, and the aversive stimulus would be something like, like an electric shock, and then they would be inside of a scanner and they would scan their brains, and then you would look at what happens. But again, we're missing the context. Here we go. And the context is what we're trying to do with this fellowship. So some of you might be familiar with what the V simulator is. It's a VR facility that exists here at Exeter that has a platform that moves, and alongside a VR headset, you can virtually recreate any scenario that you may imagine. And you can also add some wearables to it. So You can add FNIR, so you can add this gap here that allows us to kind of like track fluctuations in brain activity while the person who is moving around in this platform doing things that are more like ecologically valid, that are more realistic. And you also can measure things like, like preparatory poses and things like that with this insole sensors that allow you, that you can attach to people's feet, and then you can very, very objectively, that you can just track like what people are doing in the scanner. So in the platform. So putting all this together, we can start building on, creating that context. And this context in our case is going to be commuting on a train. So the scenario that we've created, participants would put their VR headset on and they would be on a train that is moving, and they would be asked to move from one side of the train to another. And I've got an example here what you would see on the VR headset when you are on the platform. They can hear it as well, and they can feel like the vibrations of the platform that, like kind of, it is quite uncanny actually. Like it just feels like you are in a train. And then that's me. And when we talk about, like when I was talking to you about, like we are going to present an auditory cue and then an aversive stimulus, but, like without providing a lot of context, it's difficult to imagine what that looks like. But this is an example of one trial where essentially we're just going to ask participants to move from one side of the carriage to another side of the carriage. And in the process of doing this, they'll hear an auditory cue that is also a train-related cue, like a train sound. And that would be followed by a jolt that would destabilize them. And, like we know from doing PPIE, we know that, patients are very fearful from like falling and walking in a moving train because of that fear of falling and because that fear that's going to trigger the pain. So that is what's going to be the aversive stimulus. And this is an example of this, where we're going to do nothing but just ask them to move from one side of the carriage to another. Hopefully that was obvious enough that there was a jolt there. But essentially, there's nothing that we are doing that people are in their own reality, in their own train, and they're just, they get, please change your seat, and then they stand up and they move from one side to the train to the other, and it has nothing to do with like the lab environment where they are. So now we can start like building up on this protocol, right? So now, yes, we have a fear conditioning protocol, but now we are able to make it a lot more ecologically valid. So this is now measuring objective information as participants are moving around the platform in an environment that they know and is familiar to them. So what we are going to do is, from this conditioning test phase and extinction phase, alongside insole sensor information, so we're going to be able to see how people, when they hear the auditory cue, are they stopping, do they keep walking, are they preparing in a certain way? We are also going to monitor heart rate throughout the whole experiment, and that's going to allow us to understand things like measures of parasympathetic tone that allow us to understand, like stress responses throughout the experiment. And they're also going to have FNIRs on their heads, which is going to allow us to track, brain activity fluctuations that might enable us to understand a little bit better, once they learn this fear, what are the neural mechanisms underlying this learning of fears. But this is not, as I said before, this is not the only reason for the gap. So we were also talking about a huge research, especially clinicians' engagement. And there would be, and I've met a lot of them, a lot of people who have like a very long flourishing career in pain research who have never spoken to a patient. We obviously don't want to do this anymore. And we have included a lot of public and patient involvement and engagement element in here. And this was our original plan, so meeting with patients with chronic back pain, and first to define the problem, then to give us feedback on their experimental design, and then to discuss the preliminary results and see what are the next steps. We have been keeping up with this plan, so we did do our first meeting with a bunch of patients, and they were really instrumental for the design of the project because the initial idea was not to make people move around the train, And then when this was presented to them, that they said that, sitting on a train or standing on a train wasn't really the problem. The problem was moving on a train that was already moving and, like having the risk of falling. And that kind of made us switch the design to tailor it to what they thought the problem actually was in the train. Last week we did our second study. So this is our team here. And they were able to go inside of the V simulator and they were able to try it. And they gave us loads of really, really useful feedback. Some of it being that, like they actually already fear turning back when they're inside the train, for example. So, like that on its own was already an aversive stimulus. So we are kind of making changes to make sure that, like we kind of don't have any more aversive stimulus other than the one aversive stimulus that we want. But they've been absolutely instrumental. And if people are doing studies out there and they think that they're wondering if they should do PPI or not, I would say please do it. And the BRC has been super helpful to help me get it set up. But they are going to continue with us throughout the experiment, and we just want to make sure that whatever we do, it is what they need, and it is going in the direction that they need, and that's going to include applying for the next project, for the next fellowship. And the other thing that we are going to do, not only with our PPI collaborators, is also talk to people, which it might be surprising, but it also seems to be like a very, revolutionary idea, which is actually interviewing the participants that come into your project before they leave the room. And again, this is something that we usually miss when we do field learning conditioning experiments, where we collect objective measures and then we let participants go, and then we don't really know if they were actually aware of what they were doing, if they were actually aware that they were learning a new field or not. And that's important for us. It's important. It has, therapeutic applications. So we're also going to do a semi-structured interview and adopt a mixed methods approach to make sure that we put whatever objective measures we collect, we put them into context. And that's all we've done so far. So we've done the design of the development of experiments. We are in the midst of getting ethics approval for us, which always takes longer than what you think it's going to take. And then the next sort of year and a half will be focused on the recruitment and data collection, engagement with industry partners to make sure to try and, whatever we can, get from these experiments. Eventually, we need to be able to, create something that actually people can do in their own house. People are not going to have a V simulator in their house. So we need to be able to find sort of versions of this that include virtual reality that we can use for assessment and for rehabilitation in people's homes or clinics. And dissemination and follow-up and applying for more money. So the rest. So yeah, that's my last reminder to mind the gap. And thank you very much to my funders, the BRC, of course, and the CPNN that are also helping me getting with the use of the VSIM. The VSIM team who are absolutely amazing. My collaborators and my lab, both here and at the GW4, all the plane labs at Bristol, Bath and Cardiff have also been super inspiring. So thank you.
Yeah, that's great. Sorry, I'm sorry. That's okay. Any questions, Nick?
Thanks. That was fascinating. Just a couple of questions. The first was, with the design of the protocol, are the participants aware of what's coming, or do they get very little information so that so they can't prepare specifically? And the second thing is just in terms of measuring heart rate, are you also looking at things like heart rate variability and some of the other things you can get from doing continuous heart monitoring?
Yeah, so that's a really good question. And this is something that we're going back and forth as well about how much we don't want to deceive people, but also, if you tell them straight away, then you kind of spoil the nature of the study. So what they are told is that they need to move from one side of the other experiment, but they need to bear in mind that, they might experience movement that, the fit within the movement of a train would be. So these little jolts would be, within the boundaries of what a train would actually move like, if they change tracks or anything like that. So they are told that they will be told that there would be some movement, so bear in mind that there would be some movement, but we can't give them much more information because, and they're also over there, I was by myself doing the experiment just to show you, but obviously while they were doing it, there would be two people with them and we would be, sort of making sure that they don't fall actually. So, yeah. And the second question was, the heart rate.
Yeah, do you look at heart rate variability? And that's also a measure of parasympathetic.
Yeah, so this is exactly what we're trying to measure. So they'll have a strap and we're going to be measuring it throughout. And then we want to get measures essentially of parasympathetic tone before, after. There's also some resting state periods before and after. So we want to look at the differences in parasympathetic tone and how that relates to maybe differences in pain thresholds as they go on, and how that relates to, the preparatory process. So, sort of trying to see how all of these different measures converge and explain each other.
Yeah, Matt at the front.
Thank you very much for your presentation. I was just wondering, given that you've got the V simulator, which has all these amazing pieces of kit, are you looking at using the motion capture and the force plate facilities so that you can start quantifying how people are moving, possibly looking at the inverse dynamics, to see whether you can correlate, possible force through the lower back and that perceived pain?
Yes, so we are using the force plate data. We are not using the body sensors. This is also for logistical reasons. At the very beginning, I wanted to use everything that I could. But we need to also bear in mind that they have resting periods in between. So, that whatever setup we do, they're going to have it for a long time. They're uncomfortable. We need to take into account the setup. And that was the part that was going to take me a long time. Also the part that I didn't really know how to deal with because I'm not an engineer. So I decided to kind of put it out for the time being, but that is very much the plan. Hopefully we'll be able to do the follow-up studies within the visa, and that is very much the plan. But yeah, we just couldn't for logistical time constraints.
Brilliant, thank you.
Yeah, I just had actually a quite similar question to that one there. If pain and perception of pain is so unique to each person, how easy do you think it will actually be to find common sort of signals between your patient cohort and things that you can actually get meaningful data out of?
Yeah. So that is a very good question. And the short answer is maybe we won't find common ones. And perhaps, the idea is to sort of stratify people based on, baseline measures, based on, the outcome measures that we get in terms of their pain thresholds, for example, to be what relates to what, more in a correlation type of manner or prediction type of manner rather than finding commonalities, because it's a really good point. And that sometimes has been, generalizing something that can't be generalized is sometimes I say, yeah.
Are there any questions from anyone online? Can't see any hands up. No? Quick one, Chris.
Just a quick one. Thanks, Sonia. I was just wondering, particularly with the semi-structured interviews, is there a psychological component to that? Because presumably catastrophizing is very psychological.
So we are doing a psychometric assessment first. So we are using standardized measures, standardized questionnaires to measure things like pain catastrophizing, coping mechanisms, and things like that. The actual interview is, in this case, is more focused on looking at, the ecological validity of the experiment. We want to really understand, does this relate to stuff that happens to you in real life? Were you aware that anything was happening? Because obviously we're going to be collecting measures that allow us to understand if they are showing signs of fear. But we want to understand, were they aware of that? Were they aware they were learning a pattern that perhaps was maladaptive, or were they not? And that's usually the information that we don't have, because it's really difficult to collect in terms of numbers. But then eventually you just have to go and ask people in an interview format. So that is the purpose of it.
Thanks. I think we're going to have to.
Yeah.
People can chat to Sonia again at the lunch in a minute. So yeah, we'll call it a day there. Thanks very much. And there's a comment online saying how great it is that you've got patient involvement in your study. So that's great. Okay, thank you very much. So our last speaker is Emily Plumpton. Let's just stop sharing this. How do I do this? Do that. Move on to here. Emily, hi. Yeah. So Emily's a PhD student supervised by Peter Cook, Michael Gibbons and Chris Scotton. And she's at the MRC Centre for Medical Mycology, graduated from Exeter in 2023 in biological sciences, and did a placement at GlaxoSmithKline. So a while back. Okay, great. Thanks very much.
Yeah, so hi everyone. Today I'm going to be talking to you about asthma. So I'm pretty sure that everyone here in the room would have heard of asthma. It's a really big global health burden and affects 300 million people worldwide. And it's one of the most common respiratory conditions we see here in the UK. But what you may not have known is that asthma can be triggered and exacerbated by a range of factors. And I'm particularly interested in one of these, which is fungi. So we're all exposed to fungi every day. We're all inhaling thousands of spores from the air around us. But fungi can also be a major constituent of indoor pollution. So I'm sure you've all seen pictures like these depicting super moldy houses in the news. And we know that fungi can exacerbate asthma in a subset of patients. And this relates to reduced lung function, increased hospital admissions, and asthma-related deaths. But despite its clinical burden, there's a lot we don't know. And we don't know why fungi exacerbate asthma in a subset of patients. And we also don't know how fungi are interacting with the immune system of these patients. And that's really a key question because at its heart, asthma is an immunological condition. So over the first year of my PhD, I have so far been trying to better